Okay, thank you very much, Christian, and I'm delighted to find out about this group and to participate. So thank you for the invitation. As well, yeah, talking here on biochar-based seed coatings, and particularly looking at applications for aerial seeding in forest restoration. Let's see. Okay, so you are very likely to have seen some press coverage of of this kind of thing. There, certainly here in North America, there was kind of um, uh, some hype that was covered, I guess, pretty broadly, and maybe reaching a peak 2, 3 years ago. Um, that there's been very large investments in this area that actually have not necessarily played out very well. So there's serious challenges in terms of uh, getting the drone systems to work efficiently, particularly in remote locations. Uh, and in this whole area of seed coatings and means of disseminating coated cheese, there's a lack, coated seeds, there's a lack of peer-reviewed studies. Um, it should also be pointed out right from the beginning that seed coatings are in really broad use in conventional agriculture. This is a multi-billion-dollar industry. But there's also in that area a lack of peer-reviewed publications. Those kind of recipes for seed coatings by major uh, agricultural firms are mostly closely guarded trade secrets. And yeah, so it's something that is broadly considered to be quite effective, but there is this kind of mismatch between the application and the peer-reviewed literature. Uh, right. So here is seed coating process in action. Um, so you can produce hundreds to thousands of coated seeds in minutes, even with small scale kind of apparatus. It's possible with this kind of simple coating pan apparatus to produce varying thicknesses of coatings. We're been working recently with mixed conifer slow pyrolysis biochar for this purpose, and working also to kind of optimize the particle sizes for the process, and as well, yeah, for the response, obviously. The In the industry as it exists now, you need a binding agent for this kind of process, and the most broadly used binding agent is polyvinyl acetate. We'll get to that point in more detail. So, uh, a very first point is that there's a big literature, obviously, on biochar effects on crop growth, and it should be pointed out, and I think this is kind of insufficiently represented in the literature, that there's really different mechanisms that pertain to the germination and seedling establishment stage than to plants at later stages of growth. And understanding those mechanisms is really critical in thinking about biochar-based seed coating. So it's more about um, breaking of seed dormancy, allowing seeds to imbibe water, avoidance of ice crystal formation is very important in northern climates, avoiding dry conditions that will kill off seeds. Potentially, there's very strong interactions between fungal communities, mostly negative, so things like Pythium, damping off fungi, and ion regulation is very important in that early stage. And my impression in this area is that there's not sufficient attention to these mechanisms at that seed germination and seedling establishment stage. Um, so far. There's pretty good reason from empirical studies to think that there are pretty large benefits to be had on these processes with biochar-based seed coatings. This is a paper I published a couple of years ago. I think this is still the only field study, at least with northern tree species, to be done in situ, right? So use very small kind of tea bag sized experimental units, exposing different species to biochar through overwintering, and got more than a doubling on average in radical extension, and some increases in germination rate in some species as well, but really big heterogeneity across species. Another of my lab's recent studies, this is looking at natural pyrogenic carbon in northern forests, and the kind of subsequent growth of trees. And you see, really big increases as you go from no pyrogenic carbon on the soil surface to, say, amounts that are equivalent to 50 to 60 tons per hectare, but then declines thereafter. So you can have too much pyrogenic carbon, and that I'll note that graph there is on a log scale. So those are about five-fold increases in tree growth in Jack pine. Right. So we think that there's uh, big advantages. I'll also point out on the soil surface there, it's always the case with sort of burned over sites that the pyrogenic carbon that's just deposited on the soil surface is transported around. So it tends to be lightweight and floats, and so you get movement and deposition. So it's very heterogeneous. That'll come up later. Okay, so here are some initial biochar seed coating results. And right, we kind of initial round using the industry standard polyvinyl acetate as a seed coating agent. We had a series of lab studies and found overwhelmingly negative effects. But we took those same coated seeds and ran a greenhouse study and found very strong positive effects. We couldn't attribute this to a water table fluctuation. So we were wondering what is going on here. A thing that we felt really compelled to look at is direct phytotoxicity of the binding agent. So polyvinyl acetate in kind of dilute solutions for many species is potently phytotoxic. And looking in the published literature on this, there's a dozen or so studies now out on biochar-based seed coatings. Polyvinyl acetate, because it's broadly used in the industry, has also been used in peer-reviewed studies. And there is pretty, compelling hint there. If you use other binders, you will get positive effects. If you use PVA, you will tend to get negative effects. So that's a wide-scale pattern. What's going on? There's the potential for physical kind of binding of seeds and prevention of imbibition. That's not what's going on in these kind of dilute solution exposures. So one thing so polyvinyl acetate, right, is a main component of white glue or wood glue. That's considered non-toxic, but it turns out that there's been no published work on its toxicity from kind of standard trials. And one thing that is known is that will be hydrolyzed and produce acetic acid in the presence of strong bases. And given the basic chemical qualities of biochars, this is very likely to happen. You do find acetic acid also in biochars, and it's one of the more phytotoxic elements that are in there. So here's just a comparison between kind of standard assay results for acetic acid versus acetone. So it's toxic at levels of say less than 10 millimolar. Uh, right. So it's produced, uh, in that combination. We also find that if you uh, leach the coated seeds after the coating process, that you basically alleviate that uh, inhibition that you find with the PVA binding agent coat. So that's also consistent with this chemical mechanism rather than a physical kind of mechanism. So very clear recommendation, stay away from the PVA. There's lots of other binding agents which are preferable, and we've shifted entirely to looking at a range of other binding agents. Some of the binding agents that have very low toxicity include starches or modified starches, and there's a number of kind of bio-based binding agents that come out of the wood products and paper processing industry, as well as things like hide glue. Um, I'll just point out this is a bit of an aside in this, but most large-scale biochar production facilities produce large quantities of very fine particle sizes as part of their, something that comes out that's not really intended. So here, for example, this is BC Biocarbon is one of our industrial partners. They're becoming kind of the largest producer now in Canada. Some new plants being set up, and their process, they get something like 20 to 25% of their product that is less than half a millimeter. And right, this is a problem and challenge for kind of health and environmental concerns in its application. And so they're looking really seriously at forming aggregates, granules, or pellets as part of their product mix. And that gets you squarely into this issue of binding agents. Um, uh, Also, we have kind of a series of studies that in a variety of applications we've found superior effects of granules or pellets on uh, plant performance in different kinds of applications. So here's a green roof application. There's a series of recent studies that have come out, and we find improved plant responses, improved water quality responses from the discharge from these systems, and also reductions in erosion, not only of the biochar, but of the other parts of the growth medium. So there's a variety of kind of interesting benefits of pelletized or granulated biochars, and the seed coating kinds of processes go directly with that, right? Because if you're using biochar coated seeds, you're going to end up with these kind of biochar aggregates. Okay, this is a very recent, our first large-scale field trial. This was put in last year and surveyed this summer. This involved about 1/3 of a million seeds over a 3-hectare area. This was with polyvinyl acetate as a binding agent. And right, there were a series of conifers that were used, and then also a couple legume species that were used as companions. And our results from this initial large-scale trial, the conifers was a sad story. There were kind of extreme um, heat conditions. There was a May heat wave that killed off most of the seedlings. So every year has its own kind of story. But there was, in neither the control or the biochar coated plots was there really a appreciable conifer establishment. But one of the lupins did tremendously well. in with the biochar coated seeds. So about a 20-fold increase in seedling establishment in the field in this kind of harsh environment. And that was clearly a seed germination response and not a growth response. So we got not any difference in the sizes of the seedlings that were produced, but big differences in germination. Um, uh, Set of lab trials here. This response, at least with the PVA binding agent, is specific to lupin. So other species are basically not showing any positive, it's a dramatic negative response, but lupin goes crazy. Uh, this kind of thing, it must be some germination signaling kind of process, and the most likely suspect are characins, which, if you're not familiar, are these heterocyclic compounds that are produced by combustion processes. They're kind of isolated from wood smoke. So there's a whole set of species that will germinate in response to chemical cues from wood smoke. These are broadly present in trace quantities in biochars, and so we strongly suspect that this Arctic lupin has this physiology. Uh, okay, so here's some very recent. This is an ongoing trial. So we've tried out this set of different binding agents, and cassava starch turns out to have pretty good properties. And we're looking here also at biochar with a mixtures of deactivated yeast to provide kind of early uh, nitrogen boost. That's particularly important in northern environments. And yeah, so we're seeing benefits of both biochar and yeast in uh, some cases, and pulling out the kind of mean marginal effects. Those are pretty large, so that it seems to be an additional boost from the yeast as well. I'll Yeah, there's a whole story on the benefits of deactivated yeast and its broad availability for use in this kind of context and with biochar, but um, that'll have to wait. Okay, so some take-home messages from this work. Um, Biochar-based seed coatings are really promising, but it's kind of obvious, I think, from this overview that considerable work is needed to develop recipes that are broadly applicable. And I choose the word recipe carefully. I find myself doing a lot of lab work, which we're working with these starches and so forth, and I need to look for students who have kind of backgrounds in food industries or preparation or confectionery. Also, those seed coating pans are actually purchased from makers of devices for chocolate purposes. Um, Mechanisms for biochar effects on seed germination establishment are really distinct from those of later plant stages, and we've got to understand more about those processes and the physiology involved to succeed in this area. Um, I think this is really important, not just for understanding this kind of application, but also the broader ecological impacts of biochar use in the field and of natural pyrogenic carbon. And some concluding thoughts, just I guess, especially on industry role in this. So this kind of boom and bust of drone-based seeding companies. I mean, in Canada, there's a couple of these companies that got millions, in some cases, tens of millions of industry funding, and then had such poor results that they changed their names to not emphasize the use of drones at all. And it's even the case in British Columbia that the government paid some of these companies to do this drone based seeding work that was completely unsuccessful. And so BC has banned drone based seeding operations. They can't use government funds for for this work at all. I think that's completely premature. And that's basically having these companies that have done a good marketing job that don't have their ducks lined up at all. There's critical but complicated problems to solve to make this work, but I think it's actually necessary. My time in the field has convinced me that. So I have this. This is an increasingly common situation across northern Canada. I mean, this is of course receiving international news coverage. Very hot fires coming through, to which the fire adapted tree species are not able to kind of function and regenerate through these hot fires. So you end up with ghost forests where the seeds have all been destroyed, uh, and there's no tree regeneration at all. Those are too dangerous to send in planting crews. They're in remote sites. And I don't know what else can be done to regenerate this forest other than use of drone-based seeding. Uh, so working with companies in a way that makes the academic work, uh, you know, broadly available and transparent is necessary, to convince government agencies and so forth that this is something that can work. Uh, Lots of people to thank, in particular Esther Tang. Some of this work will go into her PhD thesis, but many people who've made this work, and especially our partners with Seed the North and BC Biocarbon. And I'd be happy to take any questions. Thanks.
Thank you very much, Sean. That was very interesting and also a lot of new information for me as well, because it's a more exotic use of biochar, I guess. My first question would have been is so far you basically found that the carrageen was responsible for the germination improvement. But did you see any other effect of biochar or of different biochars? I'm not sure if you use different biochars, like a nutrient provision or anything like that.
Um, well, that incredibly strong germination response that we see in the Arctic lupin, we think there must be some kind of germination cue involved, and so we suspect that that's Carrikins. We have not uh, done all the experimental work necessary to pin that down. Uh, right. So that's kind of next steps in that work. But yeah, I mean, uh, provision of potassium, right? Of the ions that are transported for supporting radical extension growth. Potassium would be of kind of premier importance, and all biochars basically have quite high potassium concentrations. So that's clearly and likely to be an important mechanism, and also retention of water close to the seeds to support imbibition are the likely mechanisms that pulling apart exactly. You've got, like most biochar work, you've got multiple mechanisms that are involved. So pulling apart those, particularly in the context of field trials, is challenging.
Yeah, I can imagine, especially the sizing of your field trial was quite impressive. One question I had on the technical matter of when you actually coat the seeds is how much biochar is actually how much or how big is the coating of the seed? How
That
can
can be controlled by the coating process,
Mhm.
right? So you can basically put multiple layers. Uh, so you go everything from uh, tenths of a millimeter to quite large aggregates are possible. And so we do have shown you kind of a sample of the work here, but it's likely that there are optimal coating thicknesses. But it's essential to get the binding agent right. If you have a phytotoxic binding agent, then you've got to solve that problem first.
Yes, I was surprised. So the industry is still using PVA or not?
Yeah. I mean, as I mentioned, the larger agricultural industry, uh, their seed coatings are proprietary and they don't share that information. So you have to, be doing sort of back engineering and a lot of chemistry to figure out what they're using. But I do know uh, the largest seed coater in Canada has used PVA for years, for 30 years. And that's their primary thing. I think it's kind of known that their coated seeds don't show as good of germination in general as non-coated. But this is, PVA is cheap and easily accessible and works really well for the physical process.
Okay, so cost effective, I guess. Tom Miles was asking, is there value in coating with wood vinegar? So the
Uh, yeah, I think I did refer to and showed a couple of results. Um, uh, have a paper in the journal Biochar that's under review at present that is looking at uh, kind of phytotoxic versus hormetic responses to the wood vinegar constituents that are absorbed into biochars, right? So all of those volatile fatty acids from acetic acid up to caproic acid, and some more exotic ones, as well as other kinds of constituents. And, you know, uh, the story's a little bit complicated, but, basically, though, of the common mobile organic compounds that you find in biochars, those volatile fatty acids are the most phytotoxic. Um, there's especially a 2-ethyl butyric acid, which is the most phytotoxic of all of those. Uh, and they're much more than things like benzene or toluene or acetone. Uh, um, the but some of the long chain volatile fatty acids do also have hormetic effects at very low concentrations.
okay, so it will be
So
again.
you can find these benefits. I was very skeptical that there's any sort of real direct. I mean, there's benefits from use of wood vinegars if you're controlling pathogens or pests, obviously. And there's kind of uses of wood vinegar for those purposes that go back, hundreds of years, literally. Uh, but direct benefits to plants that are not mediated by some kind of pest or pathogen is more of a contentious thing. I was very skeptical, but in fact, we did find them, but they're not very big.
Okay.
You have to have very specific concentrations to see those kinds of effects.
So another case for a trade secret if you find it, I guess.
Yeah,
Um,
yeah.
so
I mean, this is
uh,
another one of these cases where biochars are always more complicated than you think.
yeah, that's for sure. Um, so Tom had a follow-up question also. If you know that, why does seed balls Kenya work with acacia trees? If you,
Mhm.
I don't know.
There is a couple publications on uh, biochar effects on seed germination in acacia. So there, the yeah, I have seen some things that also look positive, and I think um, uh, hard to. I mean, there's about 1,200 species in the literature that have where characins have been found to be germination triggers. And legumes commonly show that, so I think that there are some acacias that would be in that category. But yeah, you'd have to do some digging through the literature to document that.
So we are almost on time, but the
Great.
last questions here are from Hannes Salmi. So he or she was just mentioning that similar tests were done here in Finland. So you will find his email in the chat if you're interested
Okay,
in
great.
your results. Camilo was asking, have you tried an activated biochar using composting and then coating the seed? So basically what some people call activating the biochar prior to the coating, or do you use pristine biochar?
This is a really good question and really good suggestion. We have some chars that we're weathering and that's really likely to reduce those mobile organic compounds in general. So yeah, I mean, I think that is clearly something that could be beneficial. Um, the you know um, that combination of compost plus biochar does not always have positive effects. So yeah, it's unlikely that you would have compost as a kind of viable part of the mixture in seed coatings, but other organic compounds and that kind of pre-processing through uh, sort of natural aging or accelerated aging is a good suggestion. You have to be, there's a balance there. If you can lose a lot of the inorganic ions that may be important. Uh, so it's kind of a microbial process that would not involve a lot of leaching losses could be the best kind of uh, preserve the ions, but get rid of some of the organic compounds that are problematic, and potentially increase available nutrients for early growth would be so I guess plant growth promoting rhizobacteria in the mixture with yeast, which is oftentimes used as a carrier for those kind of bacteria, is certainly an important thing to be looking at.
Great. So we are on time. If you have a one sentence answer on the last question from Yale Yannick, what do you think biochar is the effect of biochar on seed priming?
That's another good question. Uh, defining exactly, I mean, priming things are in general treatments that would be used in when seeds are in the dormant stage. And but there has to be some physiological activity for the priming to really be careful. So defining what's priming versus what is, involved in those earliest stages of kind of activation and inhibition is a bit tricky. So I think that there's some overlap between these kinds of things and then what would be strictly considered a priming mechanism. But yeah, that's another area where there's a real research gap for sure.
So I see from your answer there's a lot of research areas still to be covered by your group, I guess. So if anyone is interested, just write a follow-up email to Sean.
Yes, please. I'd be happy to make connection with any other seed coders.